Hello, everyone. Thanks for joining. We will give everyone a moment to get logged on. Thanks for joining. We will begin in a moment. All right, let's go ahead and get started here. Go ahead and advance to the next slide. Hello, everyone, and thank you for joining us for tonight's webinar as we discuss AFTX-201, a next generation one-time genetic medicine for BAG3 dilated cardiomyopathy. My name is Nikki Butler, and I am the Director of Digital and Social Media for the Dilated Cardiomyopathy Foundation and the Genetic Cardiomyopathy Awareness Consortium. We're so glad you can be with us as we discuss this important opportunity. We have over 180 registrants for tonight's discussion. And next slide. Before we begin, I do have just a few quick housekeeping notes. All attendee microphones are muted so that everyone can clearly hear our presenters. We will have a dedicated Q&A session following tonight's presentation. Please feel free to submit questions at any time using the Q&A feature within Zoom. Please note that our presenters cannot provide individual medical advice. If you have questions about your own care, we encourage you to speak with your physician. Many individuals with genetic cardiomyopathy may benefit from seeing a heart failure specialist at an academic medical center. We also ask that questions remain focused on the topic of tonight's presentation so that we can make the best use of our time together. And next slide. We do have a poll for tonight. So Justine, if you can push forward poll number 1. We would love to know who's attending tonight's webinar. Are you best described as a cardiomyopathy patient, a family member or friend, a medical professional, or an industry professional? Thank you. It's always interesting to see the breakdown on these results. We have about 11% cardiomyopathy patients, 19% supportive family members and friends, 26% medical professionals, and 45% industry professionals. Thank you. And I want to let you know that this webinar is being recorded and it will be posted on our YouTube channel following tonight's discussion. As registrants, you will receive an email with a link to the recording. Our YouTube channel also houses a library of our previous webinar recordings, and using the auto translate feature, these videos are available in 54 different languages. Next slide. I'll share just a brief history. The DCM Foundation launched the Genetic Cardiomyopathy Awareness Consortium, or GCAC, in 2023, after recognizing a major gap in awareness around genetics and cardiomyopathy. We also realized these conditions couldn't be approached in silos. Many genetic mutations overlap across cardiomyopathy diagnoses, making collaboration essential. GCAC was created to bring patient organizations together to increase education, awareness, and support across all forms of genetic cardiomyopathy. Next slide. In just a few short years, GCAC has grown from 8 participating organizations to 11 patient advocacy groups working together alongside the DCM Foundation. That growth reflects the increasing need for education and awareness around genetic cardiomyopathy and the power of collaboration in addressing it. We are incredibly grateful to each of these organizations represented here for being a part of this effort. And next slide. We're excited to have 2 presenters with us this evening. Our first presenter, Dr. Hideo Makimura, Chief Medical Officer at Affinia Therapeutics, where he leads the clinical development of the company's innovative gene therapy programs. He is a physician scientist and biopharmaceutical executive with more than 20 years experience advancing novel therapies for cardiovascular, metabolic, and rare diseases. And joining him will be Dr. Arvind Bhimaraj, an advanced heart failure cardiologist and physician scientist at Houston Methodist Hospital. He specializes in heart failure, heart transplantation, and mechanical circulatory support, and is actively involved in research aimed at improving outcomes for patients with advanced heart disease. Thank you both for sharing your expertise with us. And at this time, we'll bring Deo on to present. Thank you so much.
Thank you very much for the introduction, Nikki. It's my pleasure to discuss AFTX-201 with everyone. AFTX-201 is a next generation, one-time investigational genetic medicine currently under development for people with dilated cardiomyopathy due to mutations in BAG3. Next slide, please. As Nikki mentioned, my name is Hideo Makimura. I'm the Chief Medical Officer at Affinia Therapeutics, and I'm honored to be here today with Dr. Arvind Bhimaraj, an advanced heart failure cardiologist and physician scientist at the esteemed Houston Methodist Hospital. Next slide, please. So before we begin, some important reminders. First of all, AFTX-201 is an investigational medicine for BAG3 dilated cardiomyopathy. AFTX-201 has not received marketing approval by any health authorities yet, including the US FDA. Second, enrolling in a clinical trial is a very personal decision, and it should be done in collaboration with the principal investigator of the trial. The principal investigator of the trial will explain the study and any potential risks associated with being in the clinical trial with you. Lastly, the information in this webinar is for educational purposes only. Next slide, please. So this is our agenda today. I'm going to begin this webinar by sharing with you who we are at Affinia Therapeutics, and I'll talk briefly about the root cause of BAG3 dilated cardiomyopathy. And I understand this was discussed in detail at an earlier webinar, so we'll keep this part very brief. I will then discuss how we designed AFTX-201 and what makes this so special, followed by where we are today in the path of drug development with AFTX-201. And then lastly, I will share some of the scientific experiments we have conducted with AFTX-201. I'll then hand this over to Dr. Bhimaraj, who will share with you some information about Houston Methodist Hospital, the current treatments available for patients with BAG3 dilated cardiomyopathy, who is eligible to participate in the UPBEAT trial of AFTX-201 in patients with BAG3 dilated cardiomyopathy, and where the UPBEAT trial is currently available. Then we'll move to the Q&A section, which will be moderated by Shauna from the DCM Foundation. And we'll start with some frequently asked questions about AFTX-201 and our approach, and then moderate an open Q&A with the audience. Next slide, please. So who is Affinia Therapeutics? Well, we're a biotechnology company developing rationally designed gene therapies. Our purpose is to make lasting positive impact on the lives of those affected by devastating diseases. We do this by reimagining the building blocks of gene therapies to create the next generation genetic medicine with the aim of improving safety and providing meaningful benefit to patients. We're a passionate group of experienced scientists and biotechnology leaders, and collectively, we've successfully led more than 50 investigational new drugs into clinical trials and secured regulatory approvals for treatments in more than 20 diseases, including in other gene therapies. Our focus right now is on developing AFTX-201 for people with BAG3 dilated cardiomyopathy. Next slide, please. So the underlying root cause of BAG3 dilated cardiomyopathy is a mutation in the BAG3 gene. The BAG3 gene produces the BAG3 protein, which is needed to maintain healthy proteins in our cells, especially under stressful conditions. This is particularly important for muscle cells like the heart, which are constantly contracting and therefore constantly under stress. So BAG3 protein is needed for the muscle cells to contract properly. And mutations in the BAG3 gene results in lower amounts of the BAG3 protein. This leads to accumulation of misformed proteins or unhealthy proteins, and that affects the muscle cell's ability to contract properly. As a consequence, the heart, which is a muscle, is unable to pump blood effectively, and this ultimately leads to what we know as heart failure. The symptoms of BAG3 dilated cardiomyopathy typically start in the 20s and 30s, and most patients are diagnosed between 20 to 40 years old. There are approximately 30,000 patients in the United States with BAG3 dilated cardiomyopathy, and unfortunately, about a quarter of these patients progress to heart transplant. Next slide, please. So we specifically designed AFTX-201 to address the root cause of BAG3 dilated cardiomyopathy. And we do this with a gene therapy that has been engineered to optimally deliver a functioning BAG3 gene to the heart. And this should increase BAG3 protein levels in the heart and allow the heart cells to contract properly. And this should lead to improvements in heart failure. Next slide, please. So the key components of gene therapy are the capsid, the transgene, and the promoter. The capsid is an envelope that carries the correct gene. It's a carrier. And we spent years designing and developing an engineered capsid to reach the heart. Our engineering is essentially a tag that allows the capsid to get to the right place, in this case, the heart. Conventional gene therapies typically don't go to muscles like the heart. So they have to use high doses to get the correct gene to the heart. But with this engineered capsid, we can specifically target the heart and deliver our cargo. And that allows us to use doses that are 5 to 10-fold lower than conventional capsids. And we believe this will lead to greater safety for our patients. The second part of gene therapy is the transgene. This is the gene that performs the biological activity that is missing or that is mutated. And in our case, we're using the fully human, full-length functional BAG3 gene. The third part of the gene therapy is the promoter. And this is the control element that tells the transgene where to turn on. And we use a muscle-specific promoter so that the BAG3 protein is made where it matters, in the muscles of the heart. And we spent years designing and developing this gene therapy specifically for people with BAG3 dilated cardiomyopathy. Next slide, please. So typically for gene therapies, teams spend about 1 to 3 years designing the capsid, the transgene, and the promoter. Then they spend more years conducting experiments in test tubes and in animals to see if it's safe and effective under these experimental conditions. Then, after regulatory review, we can finally take the treatments to patients, where we start with small clinical trials, where we evaluate 2 or more doses, and then move to larger pivotal clinical trials, and ultimately to marketing approval, where we can offer the potential treatments more broadly to the community. After many years, we've completed the gene therapy design and preclinical testing, and now we're in the phase 1, phase 2 clinical trial phase. We're now here where the green bar is. So we've made significant progress. Next slide, please. During these years, we followed best practices to make sure that AFTX-201 is working as designed. We conducted experiments in test tubes called in vitro experiments, as well as in animals like mice and monkeys. We designed the experiments with best practices in mind so that our data we generate is meaningful and believable. We had predefined scientific questions. We used comparators, what we call placebo, to make sure that we're not seeing random effects, but that the findings are related to the actual treatment itself. We used objective measures to make sure we aren't biasing or misinterpreting the results. And we repeated the experiments across multiple studies to make sure the results are reproducible. And we presented our results at the best external scientific conferences and to the most rigorous audience, the regulatory agencies. Next slide, please. To give you a sense of some of our work to date, in the in vitro studies and animal studies, we demonstrated that our engineered capsid outperforms a conventional capsid called AAV9. meaning our capsid was able to produce more protein at the same dose as AAV9. We've also completed animal experiments in mice where we showed that AFTX-201 was well tolerated, that it produced the BAG3 protein in the heart just as designed, and that it completely corrected the heart function and structural changes of dilated cardiomyopathy. We also showed that our gene therapy was better than gene therapies using conventional capsids like AAV9 or AAV-RH74 capsids in the same mice models. We also completed monkey experiments where we showed that AFTX-201 was well tolerated and that it produced the BAG3 protein in the heart just as designed. And we used these experiments to help pick the doses for the clinic. And we were looking for two doses that are both safe and with the potential for efficacy. And you could see some of our work that we presented at the American Society of Gene and Cell Therapy, the leading gene therapy conferences in the world, on our website. Next slide, please. And here's some of the data. These are images from a study that we did in mice. On the two images on the left, you have a mouse with dilated cardiomyopathy who did not get our treatment. And on the two images on the right, you have a mouse with dilated cardiomyopathy who did receive our treatment. The cross-sectional images are microscope images where we stain for the BAG3 protein with a brown stain, and the black and white images are echocardiograms or ultrasounds of a mouse heart. In the mice that did not receive treatment, on the far left, you see very little staining for BAG3. And the echocardiogram just to the right of it shows a flaccid heart with reduced ejection fraction. But in the mice that did receive our treatment, you can see robust brown staining for BAG3, meaning our gene therapy caused the BAG3 protein levels to increase. And the echocardiogram shows a more vigorous heart with a normal ejection fraction and thickened, more muscular walls of the heart. Next slide, please. We also assessed the safety of AFTX-201 in monkeys. The microscope image on the left, again, shows robust staining for BAG3 protein in the heart in brown, meaning our gene therapy caused the BAG3 protein levels to increase. In the table, we're showing results of ALT and AST, which are liver enzymes that are commonly measured to assess for liver safety. And the liver enzymes at both doses were completely normal, suggesting AFTX-201 was safe in these animals. I should also mention that we did not use any immunosuppression in this study, and the monkeys did well without any safety concerns. The monkeys looked well, they didn't have any signs of heart or muscle damage, so lab tests were all normal, and even on microscopic image analysis, didn't have any concerning findings. So all the years we spent designing and engineering the capsid produced the next generation gene therapy that can target the heart at 5 to 10-fold lower doses than conventional capsids, with the potential for greater safety and efficacy. Next slide, please. There are, of course, multiple examples of other gene therapies that are in clinical trials or have received regulatory approval. In total, more than 5,000 patients have been treated with different gene therapies in clinical trials or after marketing approval. And here are some examples of the gene therapies that have received approval in the United States, really showing the promise of gene therapies to help patients in need. I'll now hand this over to Dr. Bimaraj to move to the second part of the agenda. Next slide, please.
Thank you. Thank you very much, Dr. Makimura, for setting the stage. And I want to first off really congratulate you, Affinia, and then thank the DCM Foundation for pushing this mission of genetic underpinnings of cardiomyopathy. As a clinician who takes care of a lot of these patients, the past has been devastating to sit there and say, we know there's a problem, we're going to put you on pills and hope for the best. So I think correcting the genetic underpinnings is really exciting, and I'm honored to be part of this trial, and we're excited to be the first site, hopefully activated soon. So the next few minutes, I'll spend some time just talking about the trial and our institution. So I'm in Houston, in Texas, and at Houston Methodist Hospital, where we're a large tertiary center with a cardiomyopathy program and heart failure and transplant. As much as I am certified as an advanced heart failure and transplant cardiologist, my mission is to hopefully eliminate that segment of my job so I don't need to offer transplant for a lot of the heart cardiomyopathy and heart failure patients. So we spend a lot of time in what we call as secondary prevention in making sure people don't move from the early stages of cardiomyopathies. Next slide, please. So when you look at BAG3 cardiomyopathy as a part of a mixture of dilated cardiomyopathy, and I know many of you on this webinar are sophisticated and have been exposed to the reality of cardiomyopathy one way or the other. And as you know, when the symptoms are onset, these are symptoms of heart failure or inefficiency of the heart, which includes the shortness of breath, leg swellings. And interestingly, in clinical practice, we always see that young people don't present with the similar symptoms and gets delayed. Sometimes they have stomach upset and they have a bigger reserve. So when these dilated cardiomyopathy genetic causes get manifested in a young age, you might not pay attention to it. And of course, when we have to do the diagnosis of impact on the heart with testings, which include lab work, the echo, cardiac MRI, and then the intent of looking at what we call as heart remodeling is centered around ejection fraction, but we've also realized in the field that this is a crude number. So sometimes there are decisions that we want to make looking at MRI imaging and other markers in general in clinical medicine. And of course, the fact that if for you to have BAG3 dilated cardiomyopathy, you need to have the BAG3 gene mutation. There are medications that are available, which are generic heart failure medications, which are designed on what we call as neurohormonal blockade, which is the general principle of how an efficient heart and body interact, and we temper those down by these medications. But the bottom line of most genetic cardiomyopathies is we're kind of tempering it down and hope for the best. The underlying cause of the gene inefficiency or variation or mutations is not addressed. So, and depending on what kind of gene you have, some last longer, some at some point you break through the medications. So end of the day, of course, as you're taking these meds and do everything right, and we do everything right, in some severe cases, unfortunately, you progress to needing a mechanical support like a LVAD or a heart transplant. Both of those we call as replacing the heart. And the goal of all these efforts that many companies have taken up is to abolish the need for replacing the heart if you can salvage the heart. Next slide, please. So the trial design has been meticulously thought out by the Affinia team, and this slide kind of almost undermines some of the effort that has gone in as making it simplistic. But there's been multiple inputs that have been taken in, and I was going to ask Dr. Nakamura actually to reflect on the painful stakes that have been done, because this is not a loosely designed study cavalierly. So I'd let Dr. Nakamura talk about how much effort that's gone into the trial design.
Yeah, thank you, Dr. Brimraj. I appreciate the opportunity to comment on this. I won't say it was painful. I'd say that it was an interesting journey, but certainly we have gotten quite a bit of input from physicians and scientists like yourself, as well as regulatory agencies, and specifically patients as well. We actually engaged with the DCM Foundation early last year, and also with patients with BAG3 dilated cardiomyopathy to really provide input into the study design. And we actually set up a forum to share our ideas for the study and to hear their feedback. And we specifically heard requests from potential participants about making the study as patient-friendly as possible, including trying to minimize the number of procedures, the number of visits that are needed, as well as trying to minimize the immunosuppression. Of course, we have to balance all of this with making sure that we're providing safety for the patients. And we've in fact set up an independent data safety monitoring board to help us make sure that patients are safe. And we also needed to make sure that we had met regulatory requirements and that we're able to achieve the milestones and the bars that we need to clear from a clinical perspective so that we can continue to develop AFTX-201 with the intention of providing it more broadly to the patient community. And so it's been a journey. I won't say it was painful. I'd say it was interesting, but it's certainly been a journey where we've had quite a bit of key input from all of our stakeholders.
No, thank you for that. I think as you mentioned, it's a balancing act, and appreciate the thought that went into this. And then I can see that in the trial design and the endpoints and how it's going to help. Next slide, please. So that kind of brings us to the study design or the trial essentially here. Once we identify BAG3 patients with dilated cardiomyopathy, the first phase is first 1 and 2 phases. They're essentially looking for a dose estimation that has been formulated thoughtfully and with animal experiments. And I think in the first group that we're going to have a particular dose, and then based on the learnings from that, I think there'll be a group 2 with a second dose, and then finally, based on safety and efficacy and certain endpoints learned from these the first 2 phases, then the pivotal trial in the group 3 will then have a dose which is much more well informed. And of course, the initial phases of the trial, the focus is on safety because we want to make sure that it's a safe technology to make sure that it works appropriately for its efficacy. Anything to add to this, Dr. Nakamura?
No, I think you described it very well. Our goal is to make sure that we develop a safe and efficacious medical therapy for patients. And so the doses were selected very carefully, and both doses that we are proposing to use in the first part of the study, we believe will be safe and efficacious. But we need to demonstrate that into the clinic. And as you said, those data that we generate will help us develop or identify the right dose to move forward. We hope to be able to advance AFTX-201 quickly to pivotal registrational studies so that we can bring this to patients quickly. And we do believe that this overall study design is the most efficient way to get us there so that we can really bring this forward.
Yeah, I'm excited by your advancements and from the monkey data that you showed for the ability to counter the current big Achilles heel of the dosage and the immune reaction. So I'm looking forward to being hopeful if we estimated that these dose findings will actually even temper down the risk that we have seen in other types of viral vectors. So this will be great. Next slide, please. And for individuals who might not be familiar with being a part of the clinical trials, or if you have, I mean, these are just a few broad list of inclusion criteria, but effectively, if you're between the ages of 18 and 55, and you have a BAG3 gene mutation, and when we say dilated cardiomyopathy, we're going to be looking for evidence of impacting the heart to it to some degree. So there's evidence that there is an abnormality. So when we give the treatment, you expect an improvement. And then overall, broadly, in a relatively healthier state, so you can handle the medication and be a part of it. There is a list of exclusion criteria. Again, neither of these lists are exhaustive. We have a very rigorous way of making sure people meet the criteria. And the most important exclusion criteria is to one, make sure that you've not been exposed to AAV gene therapy before. And then they also look for antibodies prevalent to the vector. If you have it, then you wouldn't be a part of it. If you have a heart attack before, it probably means that your main problem is not BAG3 cardiomyopathy. So we don't want to, confound that. And then, of course, if you have a transplant or a mechanical heart, you're too advanced. And if you're recently gotten worse because of heart failure, you might be too sick to see the impact of it. So the unable to walk and the IV medicines kind of reflect being too sick, because in general, in clinical trials, we want to make sure you're not too sick, not too healthy. You have to be in the right ideal scenario. So we're optimizing the risk and the benefit of it. And then, of course, I talked about the antibodies, and anyone's pregnant, we wouldn't enroll. And if you have other acute illnesses or cancer, then we don't want to take a chance. It's important to point out that some of the previous trials needed individuals to have a defibrillator in place, and for this trial, that's not a requirement, and especially because some of the cardiomyopathy cutoffs and criteria are relatively a higher ejection fraction, so most people would not have a defibrillator at that stage. So you could still be a part of this study. Next slides, please. So what does this entail in the process for the trial? As broadly, there's an initial screening step, and I would say even before the screening step, there's something that I call as pre-screening, in the sense you don't have to come for the visits. We normally just review records and see whether you even meet the initial criteria of ejection fraction, or do you have the gene positivity. So there's a pre-screening phase. In general, if there are people out of Houston who cannot come visit, I think we can easily pre-screen before you have to come in person and get screened further with visits. And early on, the screening, once you meet the initial criteria, would then include making sure the further testings would qualify you for it. And a big part of it is informed consent. We'll talk a little in the next slide. And then once you meet the criteria for the trial, the main study period is about 1 year. The initial infusion is a one-time dose, which in our center, we're planning to actually admit the individual into the hospital. And we're very glad and happy that Affinia is supporting a lot of this effort, understanding the practicality. And that's what I meant by that. It's obvious that the trial is very patient-friendly, making it easy. Because I think I acknowledge, and Dr. Makamura knows, that it's not easy for individuals to be a part of trial and then go through all these things apart from dealing with the illness. And in the one-time IV dose, there is a need for immunosuppression, but compared to what other trials needed, that's what I'm excited about. There's lower need for immunosuppression here based on the biology that we've seen. So we will be using corticosteroids, which is not a lot of dosage compared to what we normally I'm used to in other areas of treatments. So you'll get steroids for a brief period of time, and then we phase down the steroids to make sure the immune system is not getting activated. There's a lot of focus on safety, as we mentioned, and that's why we had decided to keep individuals in the hospital for a week, where you're getting daily assessment and monitoring. And then the frequencies come down afterwards. And effectively, after the first week, there's every other day. And again, I'm very pleased that we're able to actually support those stays within, outside the hospital, within the city, where people can stay and then follow up closely. And then after that, the frequency comes down. And effectively, after a month of closer follow-up, the frequency is definitely lesser. So if there's someone coming from out of town, far away, depending on how far they are, we're estimating that a month of close follow-up might be needed. If you're not too far away, we got to figure out how people come in and go, especially after the 2-week period. And then, of course, on the long term, the follow-ups are mostly just clinical exam, and then some ultrasound exams, and then questionnaires, and maybe some imaging testing. So overall, early phase of high vigilance, which always is needed for these trials. And then on the long term, it really phases down. Next slide, please. And informed consent, and if you're aware of this process, I think the intent of these trials and a lot of my effort that I do in patient advocacies and conferences, and I've learned it's easy for us to be scientists and say, oh, we want to advance science and then put you into a trial. But I think as individuals, we want to spend a lot of time making sure you understand the potential benefit and the potential risk. And I always tell my patients saying, it is exciting to see a new therapy because you have a potential of getting benefit from this, but be cautious that that shouldn't be your only motivation because this is still a research trial and what the results are not known until the future. And you should also have the intent and motivation to be a part of the general greater good of contributing to the science. So we want to make sure in these trials that we spend some time talking about it, give the individual time to think about it, weigh the risks, benefits. And as Dr. Makimura mentioned, the regulatory atmosphere definitely makes sure that all these aspects are taken care of, and it's safety first always. And safety first, and you as an individual are in control in the sense, at any point, if you're not comfortable, you can, pull out of the study. There's no commitment, but we always want people to follow up. So you want to spend some time thinking about this and talking about it. Next slide, please. And Dr. Makimura walked through this earlier, but effectively, this is for me as a therapeutic intervention, what is fascinating and exciting is it's a one-time cure. And if this works, I mean, we're changing the landscape of not taking 10 pills, following up regularly with the pills. And effectively, the gene therapy, as mentioned before, is going to target the protein that is being affected, which is a co-chaperone protein in the heart cells, and then makes the heart cell work better. So the hope is if this works, you shouldn't have the impact of the heart getting affected by this abnormality. Next slide, please. And right now, there are 10 trial sites being planned by Affinia, and we are down here in the south, and we're excited to be the first site to get active. And I think we're waiting in a couple of days or so. We should be able to open up. We're just waiting for one or two training people to be done and checked off. And I also want to commend Affinia and thank them for making a lot of effort for this therapy to be accessible for individuals if need be travel and really engage people who need this. So we are also very fortunate to have the ability to provide this if you're traveling from outside and we have the facilities here and a lot of this is covered by the company under the trial because most of it that you're getting is part of the trial care. So we're very thankful and grateful for that. Next slide, please.
Yeah, thank you, Dr. Bhamaraj. I'd like to just echo one comment that you did make. We fully recognize that not every patient is going to be living near Houston or living near some of the trial sites that we have planned, and many patients will have to travel. And we as Affinia will make sure that you are compensated for travel and accommodation related to the clinical study so that you can participate in the study without concern for those factors. So at this point, I'd like to introduce Shauna from the DCM Foundation to moderate the Q&A. We'll start with some frequently asked questions, and then we'll open up to questions from the audience. Shauna, I'll hand it to you.
Yes, thank you so much, Dr. Makimura and Dr. Bhamaraj. Very informative. And yes, as you mentioned, we'll go through a couple of kind of frequently asked questions. We do already have some questions in the Q&A as well, and I encourage anyone who has questions, you're probably not the only one who's wondering. So please type it in the Q&A, and we'll be happy to go through those for the rest of the meeting. But first to start, the first question is, why does Affinia use an engineered capsid? You mentioned this, Dr. Makimura, in your presentation, instead of a conventional one. Can you explain that a little further?
Yeah, happy to do that. So We believe our engineered capsids really have the potential to improve both safety and efficacy of gene therapies. I mean, we have spent years designing and developing these capsids, and this particular capsid, which is called ATC-187, really targets the heart. And that allows us to use lower doses than conventional capsids that do not have any selectivity, right? Remember, if you don't have any selectivity for the heart, you end up having to dose higher and higher to make sure that the heart cells are getting the transgene, the vector. But because this engineered capsid targets the heart, we really think we're going to be dosing at 5 to 10-fold lower doses than what's been previously used in the industry. And we think that this is going to have significant benefits for safety, especially on other tissues.
Yeah, if I may add, I think as Dr. Nakamura mentioned, I was a part of the Circa 2A gene therapy, which was one of the first gene therapy trials in heart failure and cardiovascular medicine. And of course, it was not a targeted one, AAV9. When we do coronary injections, and it was a coronary injection, not an IV injection, because we wanted to focus onto the heart. Even then, the ability for this the viral vector in the genome to be taken up by many tissues, mostly liver and lung, is high because it recirculates. So I think, and I I don't want to claim to be the expert, but Dr. Makimura, correct me if I'm wrong. I think that's another advantage for me when I look at it from a clinical perspective.
That's right. It's the exposure to the other organs that will be significantly reduced by using these lower doses. We believe that will be a major advantage of using an engineered capsid.
Fantastic. Thank you. Why don't we go to the next slide? We have a few more of the frequently asked questions to go through. One, and I think, this is one that I'm a patient as well, and a lot of us as patients have questions, is how long does the gene therapy work?
Mhm. Well, I mean, maybe I'll say something here, and then Dr. Bhamaraj, feel free to comment. In our experiments with mice, the BAG3 protein from AFTX-201 was produced for the lifetime of the mice. Our intention is to develop a one-time therapy, so a one and done therapy, where we just have to dose a patient once, and it's effective for the remainder of their life. And so we certainly hope that the treatment will be permanent. But certainly I think we have our mice data to support that. Our monkey data supports that this is going to have a long-term effect. We just need to demonstrate it in patients.
Fantastic. Thank you. And Dr. Bimaraj, you briefly mentioned, what it might look like if a patient is going through the treatment. Can you elaborate how the medication is administered?
Yeah, so I think the IV, it as earlier mentioned, it is just a regular IV infusion, like it would be if you just get a peripheral IV. We're planning to have what we call as a PICC line, which will be an IV, which will stay in a little longer just to make it easy for blood draws that we're doing on a daily basis. But the plan would be to admit an individual to the hospital, get the infusion, and then there's a lot of monitoring on a daily basis for the first 7 days. And it's a hospital environment, monitoring the heart rhythm, getting seen on a daily basis, and then the blood work. And at the end of the week, our plan is to then get people out of the hospital, but we have planned to keep individuals in a hotel which is connected to the hospital. And again, I'm really grateful that Affinia is covering that cost, because after that first week, we're seeing individuals are getting lab work every other day. So it made sense to have them close by. Because one of the challenges when this is such a novel therapy, if you go back home and you end up in an emergency room, it's going to be difficult to manage. And then at the end of that second week, where the frequency comes down.
And that actually leads us to, if you go to the next slide, Justine, you'll see the bottom question is about hospitalization. So it's technically not required, but generally recommended. Can you elaborate a little further on that?
Yeah, it's not required. And I think in our experience in gene therapy trials in the past, especially when we're doing like phase 1, It's probably makes sense to do it just because there's so many unknowns and we don't want this to become a surprise. And also, if you're doing blood work every day, unless you live next to the hospital, it just is not practical, especially for us in Texas. The medical center is a place that no patient wants to go. You spend $15 for parking. Um, I think that's why we're very grateful we're able to do that. And also, when we're doing something new, we always believe in innovation, even in clinical medicine. We're pushing the realms for the betterment of our patients. But when we know we're pushing the realms beyond what we know, we have to acknowledge that we have to be ready for the unknown. And it just practically works.
Yeah. Just if I can make a comment, Shauna, about sort of the evolution of gene therapy over time. So if you look at some of the earlier gene therapies that were developed, for example, for Duchenne's muscular dystrophy or for some of the other diseases, hemophilia, for example, those studies, the clinical studies that were done to support their development did require hospitalization, and that allowed uh, both the provider, the physician, and for the patients and others to get comfortable with the medication and how it was administered. Over time, that disappeared, and there are many of these drugs that are now administered as an outpatient. And so the hope is, and the reason why this is optional, is that once we start to acquire some clinical experience and we have a better understanding of its potential risks and benefits, the hope is that this could also be an outpatient treatment. And that's why having a simple administration by an intravenous infusion is really important, right? As opposed to some of the historical treatments for heart therapy, where perhaps they might have had to do a cardiac cath to get to intracoronary injections or direct cardiomyocyte injections in the past. This makes it more uh, much more approachable, if you will, and much more available to patients more broadly. So we're hoping that this will evolve with more and more clinical experience.
Fantastic. Yes, thank you. And we do have quite a few questions coming in. So why don't we breeze to the next slide? We just have a few more here. Specifically, this is also something that's coming in the live Q&A, is about immunosuppressants. Can you speak to, Dr. Nakamura, what that looks like after the dose is given?
Yeah, so clearly you have some very savvy audience members who know what some of the other conventional AAV gene therapy capsids are using. And um, as I said before, with the conventional gene therapies, you typically have to increase the dose to make sure you're delivering enough of the correct gene to the heart. And when you do that, you end up stimulating the immune system. And so you have to use stronger and stronger immunosuppression agents to tamp down the immune system and prevent significant adverse effects. And people have tried a variety of different agents, but right now it seems as if most are using what's typically called the triple immune suppression regimen. Um, our experience is actually very different. We're using doses that are 5, 10-fold lower than these conventional capsids. And so we actually have not seen any immune activation in our animal studies, whether it be in the mice or in the monkeys. Um, we are, however, recommending that we start the study with one single immune suppression agent to make sure that patients are safe and that we are not missing anything. But we certainly think that there is a large part of dose that's actually playing a role in immune activation. And we believe that the lower dose that we are using will lead to better safety from an immune activation perspective. And that would allow us to get away with a lower severity, lower dose of our immune suppression. Dr. Bimraj, would you like to comment on that aspect as well?
Yeah, no, definitely. And I think some of the questions that will follow in the chat, Shauna, I could get specific. So just to cover this, for this trial, what is being recommended and designed is to use steroids as the single immunosuppressants. And the first 3 days, it's a dose of IV that you get once a day. And it's based on your body weight and then after the 3 doses of IV, we then go to pills, prednisone, and then slowly taper it down over a period of time until you get to 5 milligrams. And during this time, and again, I think as Dr. Nakamura said, there's a little bit of unknown, but the known is that maybe you don't need any immune system, but you want to be cautious. And as we start cut down, if there is any surprises, because the other thing we have to remember is humans are not mice, and each human is very different. So sometimes the immune system is very different each individual. If there's any reaction, there is an ability to add more immunosuppressants. But on overall, we're sticking with steroids. And in the scheme of things compared to other studies, that is very conservative. One of the drugs that has been typically used in this situation is sirolimus. I deal with heart transplant patients, and I know that sirolimus is not an easy drug to handle. It's tough. 1 in 4 don't handle it. So I'm glad we're able to get away and not need those medications with higher morbidities.
Thank you. Yes, very informative and does directly answer some of the questions that have come in. So I appreciate that. And the last question that we wanted to proactively address was about future care. So if you go to the next slide, specifically about someone who maybe their disease continues to progress and they require an LVAD or a transplant. Is there any contraindication to that, given that they might have participated in this trial?
No, I can take that up, and I think initially, as I said, when we did the Circa-2 trial with the AAV9, there were some concerns. Oh, will the viral vector have some problem for immunosuppression after transplant? And I think a lot of those were unfounded. So at this stage, I think we know enough that the gene therapy would not disqualify you for either an LVAD or a heart transplant.
Great news. Thank you very much. So with that, we're going to show a brief video. Many of you on the line have attended one of our sessions before. So we'll do that first. Dr. Nakamura will walk through what to do next, which was another question that came up from a few patients here of, maybe I'm interested, what should I do about that?
Yeah, so that's great. So this is our call to action slide. So if you are somebody with BAG3 DCM, or you are a family member of somebody with BAG3 DCM, or perhaps you're simply just more interested in learning about the UPBEAT trial, I would urge you to reach out to us at our email, clinicaltrials@affiniatx.com. Our patient advocacy lead, Kristen Smith, can help answer questions or guide you to the right place, including potential clinical study sites. If you would like to speak with Dr. Bimraj's team at Houston Methodist, you can contact his team at the email and cell phone listed below. This information is also in clinicaltrials.gov now, and that is live as of yesterday, I believe. And if you prefer to speak to somebody at a third-party patient group like the DCM Foundation, you can scan the QR code. I believe this QR code will be shown later as well, but you can contact patient advocates at that link as well. So thank you very much, everybody, for your interest, for joining the call, and I'll hand it to you, Shauna, to share Greg's video.
Thank you. Yes. So I was just mentioning, if you've attended a webinar before, you're probably familiar with Greg Roof. I've had a conversation with him. He was unable to be here this evening. However, he has a message that he wanted to share and make sure that everybody hears. So let's go ahead and play that.
Hi, this is Greg Roof from the DCM Foundation. Sorry I could not be here with you this evening, but I'm attending the Global Heart Hub conference in Europe, and it's about 1 a.m. my time right now, and I have an early flight tomorrow. I'd like to thank Arvin and Hideo for their wonderful presentation. I would also like to thank Affinia and other companies that are creating genetic cardiomyopathy therapies. It gives us all hope for the future. Also, as a genetic cardiomyopathy patient, father, and grandfather, I would encourage all of you, if you have BAG3, your family member has BAG3, no matter where you are in the world, to take a look at this clinical trial and see if it meets your criteria and it's something you might be interested in. I really believe that all of us with genetic cardiomyopathy have an obligation to try to help ourselves, help our family members, and help our genetic cardiomyopathy community. Thank you and have a great evening.
All right. Thank you to Greg. And with that, I would love to transition to the questions that have come in. So thank you, everybody, for your questions. We'll probably go about 5 minutes over. So thank you to our speakers for that. And we appreciate everybody taking the extra time. There are quite a few questions related to the engineered capsid. It's drawing a lot of attention and interest. So my question for you, Dr. Nakamura, there are a few. The first two are really about whether you believe that this capsid could apply to other genes. But one example is FLNC, for example, and if you think that's something down the road that could happen.
Yeah, that's a great question, and certainly at Affinia, we believe very strongly that this capsid and its what we call tropism to the heart is special, and we do think that this could potentially apply to other diseases related to the heart. So other inherited cardiomyopathies are something that we would be interested in, and we do have a pipeline of other cardiomyopathies that we are considering. And certainly, if this is successful, we would certainly like to explore many other possible diseases that we could think about.
Well, thank you. And we have a follow-up to that actually is the kind of classic capsid have been extensively studied. Is there any concern of safety for these engineered capsids?
Yeah, it's a good question. And of course, until you get into the clinic, it's hard to know. But what I can say is that we have done everything we can to make sure that it is safe, including all the animal models and the animal studies. In addition to that, I would say is that these engineered capsids are becoming the new frontier for gene therapies. And there is actually another company that has put forward an engineered capsid that has a similar approach to ours. I won't say it's the same. I still think ours is better, but a similar approach. They recently published their clinical data, and it appeared to be very safe and, in fact, was more efficacious in terms of delivering the gene compared to conventional capsids. And so I actually think we are actually at the frontier of these engineered capsids, and we're going to see more and more of these as the field progresses.
Fantastic. Yes, thank you for that. The next is about the gene therapy not only helping DCM, but would it also help the accompanying muscle weakness?
Yeah, that's a great question. So this particular capsid is what we call cardiotropic, but it does also get into the muscle. And you might have caught me when I talked about the third element of the gene therapy, the promoter. And I mentioned that we are using a muscle-specific promoter. So there is hope that this will not just address the cardiomyopathy, but also the myopathy, the muscle weakness associated with some of these diseases as well. But of course, we have to do the clinical study to understand how much of a benefit we would expect to see.
Thank you very much. The next questions are related to inclusion criteria. So Dr. Bimaraj, maybe if you want to take this, the first one is about, is there an ejection fraction or EF criterion as part of the inclusion criteria? Because that was not listed in tonight's presentation.
Yeah, there is a ejection fraction criteria to be less than 45%. So, and I think in that regard, I want to also caution that sometimes the ejection fraction measurements can vary from place to place. So there's a 10-point variation. So if somebody's ejection fraction is 50, 55 in some forms, it could still meet the criteria. So, but we need a documentation of less than 45% for us to qualify for the trial.
Thank you. And we actually have someone who has an EF of 45, but outside of the age range, older than the age that you provided for inclusion criteria. My suggestion is, regardless, to fill out the information form and try to get more information about the trial. I see you both nodding your heads. Welcome any other feedback that you'd have or commentary.
Yeah. So as part of the team that helped design the clinical trial, I would say, we did have a lot of discussion about what the appropriate age range was for this clinical trial. And I'm not going to go and summarize many months of discussion. But what I would say is that at least for the beginning, in the initial stages of the clinical trial, the idea was to keep the population, as Dr. Brimraj mentioned, a balance of healthy and sick at the same time. And so to keep it relatively young. But of course, down the road in the future, would we consider expanding the age range to older patients, perhaps even younger patients? Absolutely, that's something we would continue to talk about depending on the clinical data we generate. So if you are interested, I would recommend that you either talk to us, talk to Dr. Brimraj's team, or scan the QR code and talk to the DCM Foundation and make your interest known. That would help us think about our future studies as well.
Absolutely. Thank you. So we'll do 2 more questions and then we'll give Nikki a couple of minutes to wrap up and again, provide some reminders. The one question is about the screening. So do you screen the patient before dosing to be qualified? Can you talk about what that screening looks like?
Definitely, I think there's extensive screening that we would do before we dose. And as I mentioned initially, there's probably a screening where we will look at the clinical criteria, make sure they meet the ejection fraction. There's some symptoms, there's some blood work. And then in the visit, we do the EKGs, we do other testings to make sure before we decide on moving forward with the dosing, there's a whole period of time that we have to make sure that we're meeting all the criteria for safety.
Fantastic. Thank you. And that's, I think, why you talked about the informed consent and, making sure that we really encourage, we heard this in Greg's message too, and I will echo that from the DCM Foundation, encourage you to reach out and find out more. You're not signing up for anything until you go through all these steps and make sure that it's the right fit for you and you're the right fit for the trial. So we encourage you to go and kind of, investigate this for yourself and your family. And then lastly, we'll get one more. Would patients remain on heart remodeling drugs and for how long until heart function could return to normal?
Yeah, so the trial will not interfere with the clinical care. If somebody is on the heart remodeling medications, then they would remain on them. And I don't think at this stage we are intending to withdraw the medications because it opens up a lot of controversies because right now the focus is on safety and efficacy. It's well known in general, whether you have BAG or not, that if you withdraw the medicines, things could get worse. And then if you're already on the meds, then we would definitely not withdraw. Whether you put people on meds or not, it's a little controversial with the ejection fraction being borderline if you're newly diagnosed. But I think the trial would not interfere with the clinical care, which will have to be decided by individual physicians and make sure that that's not being compromised.
Thank you very much for that answer and both of you for walking us through those questions. And thank you everybody who submitted a question. At this time, I'll turn it over to Nikki to wrap us up for the evening. Thank you.
Hello. Thank you all for being here and for taking the time to share and to learn more about this important research. If you are interested in exploring the UPBEAT clinical research study in more detail, we encourage you to visit our webpage. You can see a screenshot here. We also have the QR code shortcut in the URL listed. On this page, you'll find clear, up-to-date information about the study, how it works, and what participation may involve. And next slide. Also on that page, you can complete the inquiry form to have a trained independent patient advocacy expert reach out to answer your specific questions. Please know that filling out the form does not obligate you to participate. It's simply a supportive next step to help you stay informed and to make the decision that's right for you. And next slide. As stated earlier, registrants will receive an email with a recording of tonight's webinar. The email will also include a link to the UPBEAT Clinical Research Study webpage referenced on the previous slide. The Affinia contact email address will also be included and the link to register for our next webinar. And next slide. Our next webinar will be held Wednesday, August 19th. We are going to take July off. That will be at 6 p.m. Eastern Time. And our topic will be targeting the genetic root cause, Atrium Therapeutics RNA approach to rare cardiomyopathies. Registration for this webinar is currently open, and information can be found on the DCM Foundation and the GCAC websites. Remember that even if you cannot attend live, please register and you'll receive a copy of the recording via email. And next slide. Thank you to all of our sponsors whose logos you see on the screen. Their support allows us to present this important information. And thank you again to all of the patient partner groups for their ongoing support. Thank you, Justine, for your technical Zoom assistance behind the scenes. And next slide. Thank you all for joining us this evening to learn more about AFTX-201 and the UPBEAT clinical trial for BAG3 associated dilated cardiomyopathy. We're especially grateful to Dr. Makamura and Dr. Bimaraj for sharing their expertise, insights, and commitment to advancing research for the genetic cardiomyopathy community. To everyone who attended, thank you for your questions, engagement, and dedication to learning about emerging research and potential new treatment options. We hope tonight's discussion has been informative and encouraging, and we look forward to continuing these important conversations together. Have a wonderful evening, be well, and we'll see you again in August.